Okay, Chelsea has the green light. Welcome to IACS a student seminar. I'm happy to introduce Kementzidis from our group in the Department of Applied Mathematics to give a talk on the topic he presented on AI-guided management of multiple scales for MD simulations. Yogurt has been with us for 3 years and with our group for a little over 2.5, probably about 2.5, and is a very active member of the team. As you can see, we have 18 people online, and I could see quite a few in the audience. Yogurt is all yours. We'll look forward to your presentation.
All right, thank you so much, and I guess we can get started. So, my name is Yorgos. I'm a student in the Department of Applied Mathematics and Statistics, and I'll be talking about how to manage multiple scales for MD simulations with a little bit of help of AI. So I'll start with a little bit of motivation, some introduction, and then the second component of my talk will be split into two parts, like 2 chapters. Okay, so first motivation, then the specific topics. Okay, a few things. Number one is cardiovascular diseases are the leading cause of death in most of the world nowadays. And that's probably a good enough reason to study them. And well, if we want to study cardiovascular diseases, we have to start from somewhere. We choose to focus on a plasma protein called fibrinogen. Its function is it kind of like assembles, polymerizes, it creates a net, a fiber net, that serves like a scaffold for a blood clot, a thrombus. So as you can see in the diagram over there, you have this net. This is like the proteins that are assembled together, and then either blood platelets or red blood cells go there and they form a clot. So we want to study this scaffold formation, this net formation. We want to see the properties of the molecule and so on. To do that, we can use MD simulations. Now, of course, there's advantages and disadvantages. Number one, in terms of advantages, we can look with all atom molecular dynamics simulations. We can look at the atomic resolution and study high frequency phenomena. So we can have a very small time scale and look at things in detail. Well, this way we can provide some insight into flexibility, conformational changes, and so on. We can look at the mechanism, I guess, by which things are happening and not the fault behavior that a typical lab experiment will look at. Now, don't get me wrong, I'm not saying that we aim to replace in vitro experiments, but we can enhance them, complement them. So that's one good thing about simulations. Now, there are some disadvantages. Number one, if we want to have these small time scales and study these high frequency phenomena with also small spatial scales, this can become very computationally expensive, even with all the advances in high performance computing, GPUs, so on, parallelization. It's still very, very, very slow. Like for a large protein, we can have simulations that can last from weeks, even up to months, if we want to see some single molecule simulation. Now, there are some phenomena that can take that last in the orders of microseconds or even milliseconds, like protein-protein interactions or even protein folding. And in these cases, MD simulations are like even prohibitive. Like we will not get the results that we want on time. Anyway, so what's an alternative to this? Well, we're gonna do can do multiscale modeling and coarse graining. So I'll be talking about coarse graining as a methodology that we use. So in rough terms, coarse graining, you take some atoms that are grouped together, and you group them together, you represent them by a single bead. So this is like just a sketch, I guess, don't try to get the chemistry out of it. The idea is that from like 25 atoms, you can reduce the atomistic resolution to like 5 beads. So now you have fewer beads, fewer interactions, fewer computations. So now your simulation can take less time. Of course, you have to sacrifice a little bit of accuracy for this, but I guess the hope is that you're going to do it in such a wise and balanced way that you're not going to sacrifice too much accuracy. This way, now, because the simulations can take longer, you can access different spatiotemporal scales. Now, we don't just do MD simulations at the atomic level. We can continue and do simulations at the molecular level or even cellular level. So this is like a big help, a big boost in our ability to access those spatiotemporal scales and simulate a variety of phenomena. Now, the tasks that that we have to worry about during coarse graining is mapping the geometry from one scale to another, and the other task is matching the dynamics. Okay, obviously the force fields from one resolution to another are slightly different, and we can see how, I guess, one of the two tasks, force matching, can be a little bit more difficult. Now, I talked about managing scales, so I don't just want to coarse grain systems and get the lower resolution to run simulations faster. I also want to restore the atomistic resolution from coarse grain structures. So we're also going to talk about another problem. We're going to talk about inverse coarsening, like de-coarsening, backmapping. So which is pretty much the equivalent of image super resolution, but with molecules. With image super resolution, you start with a blurry image, you want to clear it. Okay. Here we start with a coarse grain structure, and we want to make an educated guess as to what the molecule is supposed to look like in the fine-grained, the whole atom resolution. Okay, this is the motivation, so let's get started, okay? So first I'm going to talk about forward coarse graining, okay? So in order to speed up the simulations. A few introductory remarks about fibrinogen. I said a few things, plasma protein, high molecular weight, elongated. One thing I want you to remember, I guess, is this sketch here, because it's simplified. The protein has an E region in the center, 2 D regions on the sides. The molecule is symmetric. It consists of like three pairs of chains. So you have the E region in the center, the D regions on the side, and then the alpha C domains being tethered in the E region. So they interact with each other and with the E region. So at first the molecule looks like this. So let's say, I guess, what happens in our body. The protein starts in this form. And suppose you have an injury or you're having a stroke. One thing, one worse than the other. What happens or what can happen is that like thrombin, which is an enzyme, goes here in the E region and kind of like releases these alpha C domains. What I'm saying here is it cleaves some fibropeptides. So it releases those alpha C domains, and now these are free to like swing by, like they're like swinging hands. They allow intermolecular interactions. So eventually, with these intermolecular interactions, we can form the fiber net. Okay? How is this formed? There are some specific interactions that take place. Just of the terminology, when we release the hands, the alpha C domains, we will be calling this an a fibrin monomer, not fibrinogen anymore. And the idea is that there are some interactions, AA no-hole interactions, between the D and the E regions of molecules that bring the proteins together. And this interaction, along with some specific interaction between the alpha C domains, the dark gray hands, lead to the polymerization. So you have some protofibril formation first, and then with some lateral aggregation, you get the bigger net eventually. Okay, so what I want you to keep out of this is that we start with a fibrin monomer, and there are some intermolecular interactions that lead to the net formation. Okay, so now this is the biology. How do we do coarse graining to speed up these simulations? Okay, so the methodology, I guess, the term we're using is shape-based coarse graining. We're pretty much, as I said, grouping atoms together that are close to each other, but at the same time, we want to preserve the topology of the initial molecule, like the bonds, dihedrals, and so on. So using a shape-based coarsening algorithm, we can reduce a 42,000 atom protein to about 15 beads. This is like very ambitious, but for the purpose of studying fiber assembly, probably our only way. We have to reduce the atomistic degrees of freedom drastically. So pretty much, not 100% true, but to some extent, we're taking the center of mass of some atoms that are close together. So for example, some atoms that are here at the very end of the protein are mapped to a bead. Okay, now, this is matching the geometry. It's relatively easier than matching the dynamics, which is the next step. So we want to learn a potential so that a potential that takes the CG coordinates as input, and it gives us, I guess, the potential of the molecule at this resolution. How are we going to do this? We're going to use a bottom-up approach, and we're going to take an all-atom MD simulation, fine grained. So we're going to run a simulation with all the atoms, and then we're going to try to form a potential that matches these forces. So if we focus a little bit on the formula here, we have the weighted average of the forces in the all-atom resolution. And here we have the negative gradient of the potential. So the forces of the forces that are computed at the new CG resolution. And the point is that we want to match these forces. Okay, so some questions that can arise is like, how are we going to determine these parameters? What are the starting points? Can you be a neural network or something? Because we are not good enough at giving meaning to models. Well, there's a lot of things that we can consider. We can have a black box implementation, like a deep neural network, but the problem with this is that the parameters that are derived are not interpretable, and the predictions can be unphysical sometimes. So instead, what we're going to do is we're going to you embed the physics into the learning. So we have a form of the potential. It consists of a variety of bonded and non-bonded interactions at the CG resolution. And our goal is to estimate these parameters. for each different type of bead. So I guess instead of like trying to compute the nodes in a neural network, we instead try to compute some more interpretable parameters, making sure that eventually the estimated potential is regularized. Let's look at the schematic a little bit. The idea is the following. You start with the snapshot of the all atom MD simulation. You take the features of this simulation. You get bonds, angles, dihedrals, non-bonded pairs, and then you make an estimate. You have an estimate already about the force coefficients. You compute the potential, and then you match it with the forces from the average forces from the all atomic simulation. And the goal is that over time, this network with backpropagation, it will like fine-tune the coefficients in such a way that they're gonna reflect the actual all atom simulation. So this is what happens. We train it, and obviously there is some training and testing. We train it, validate it, and test it on new data. And we see at first that the model matches the forces. So between the two resolutions, there is a match between the forces. But this is not always enough, right? When you want to when you want to evaluate an MD simulation, you have to look at like some other distributions and see if they are preserved across different scales. Now, the model we'll be using for our reference is called CGSNet. So it's a coarse graining with a SNET architecture, which is pretty much like a graph neural network based architecture with continuous convolutions, which is probably very good for small molecules because it's been tested on small molecules only. But I guess until we tried it. But when dealing with much larger molecules, like a 40,000 atom protein, it doesn't work that well. Probably because I guess these graph computations and message passing neural networks include very heavy computations and have a very high memory demand. Now, so we're going to be evaluating our model. We run our simulations for the physics information in LAMS. And first of all, we see a huge speed up from the all atom resolution, 92,000 times, approximately 10 to the 5. Also larger than the CGSNet. And now we also want to check if it is also more accurate. So we're looking at distributions. Number one, we're going to look at the radial distribution function. Like if I have to sort of explain it in one word of a sentence, it's not exactly this, like an idea of how density and like bond lengths vary across the system. So one thing we can see, I don't know if you can see the colors very well, is that our physics or parameter learning matches the distribution, the radial distribution function of the original simulation much better. And if we want to quantify this, this is only for one snapshot. I think this is at 5 microseconds. If we want to see this across multiple time steps, we see that the JS divergence, some sort of statistical distance between our radial distribution function and the all atom radial distribution function is much lower than the one with CGSNet. So we're not just matching forces, we're also matching some distributions. Another thing, Ramachandran plot, we want to see how the dihedrals are matched from one simulation to another. So we have the free energy landscape. And we're looking at different types of dihedrals. and we can see how this is the all atom simulation, this is our simulation, and this is with CITUSNet, how we can capture more of the dynamics of the simulation with our model. So, okay. Now remember that this model was only trained on a single molecule trajectory, and it was tested on a single molecule trajectory. Now, if we want to use this for a multiple fibrinogen simulation, we want to also incorporate some interactions that were not part of the training data set. So for example, nodal interactions. They're not part of the training data set. Remember the D and the E regions attracting each other? Well, in the training, we only had one molecule. So this thing was not there. It wasn't really captured. So we have to embed it. What we're going to do is, because we're at a higher scale, we're going to model it with some Coulomb force. The nature of these interactions is not fully understood, maybe a little bit electrostatic, a little bit hydrophobic, but we're going to model it at our scales with a Coulomb interaction that also turns into a bond if the two regions come at like a sufficiently close distance. So this is the step function here, the H. And also we want to embed some specific interactions between the alpha C domain. So if the alpha C domains, the dark gray hands, the swingy hands come close to each other within some threshold distance, they also form a bond. So, okay, let's look at the simulation now. This is at 8 milligrams per milliliter. We have some fluid, but I've made it transparent so that we can see the molecules. We're modeling the fluid with dissipative particle dynamics, and we have a time step of 7.6 picoseconds, a box with periodic boundary conditions, and we're using this counter wet flow just because it is, counter wet flow means that there's flow on one side in one direction, flow on the other side in another direction. Why do we do that? Well, we want to just speed up the process by which the molecules come closer to each other. If we don't have it, it will still happen. There will still be polymerization, but it will take much, much, much longer. Like we see it happening, but if we want to see it quickly, we have to add this flow to bring the molecules together. If you think about it, if you have an injury and you bleed, sometimes forming a clot might take a minute, sometimes a few minutes. So we're working with picoseconds, so that's still not a big speed up. I mean, it's a big speed up from femtoseconds, 10 to the negative 12, but still, we can't do it without the help of this flow. Anyway, you see that there are some clusters that are formed. There's some lateral aggregation, and there's these D and E bonds formed between the molecules. So this is so far a pretty good picture, like a pretty good simulation, I guess, of the polymerization and how the scaffold is formed. Now, if we want to do a little bit more, a few more measurements and try to quantify things, I mean, that's an advantage of simulations. You can offer some quantitative perspective. We can have a bunch of other metrics. We can look at the fiber count. What we call fiber count is the percentage of molecules in the simulation that are part of a cluster of size A. So if I have a schematic here, and suppose that each vertex is known as a molecule, not just an atom, let's say it's the center of mass of a molecule. Two molecules are adjacent if there are any intermolecular bonds between them. And for example, these molecules, H, D, and E, are part of a 3-fiber, a cluster of size 3. The ones here are part of a 4-fiber. This one is a single molecule, it's 1 fiber. So we want to see how things look like over time. So the thing. So over time, nothing unexpected. Most clusters are single, one fiber. Over time, we see a rise in 5+ fiber. Okay, nothing unexpected, 8 milligrams per milliliter. Then one thing we can do to corroborate our findings with in vitro experiments is to see how different concentrations lead to different percentages. So here we see how the fibrin ratio. So any molecule that's part of a cluster, regardless of size, how if we start with a different concentration, 8 milligrams per milliliter, we get a higher percentage of clustered molecules than 2 milligrams per milliliter. Same thing for 5+ 5, long and big fiber. So that's one thing. Average and median diameter. Diameter, maybe it's a bit confusing of a term. We don't have circles, but we also don't have graphs exactly because we are interested in the Euclidean distances. Within each cluster, I call diameter the biggest distance between any two atoms within the cluster. So if there are any two atoms inside these molecules here, this will be the diameter, this will be the diameter, and so on. So over time, if we want to quantify aggregation, we expect this number to get bigger over time, right? Like, because the idea is that if the molecules are clustered together, and especially they form elongated clusters, we expect this diameter to become higher. So we see this both for the mean and the median diameter. The median diameter stays flat here. Why? Because a lot of the molecules are not even clustered at 2 milligrams per milliliter, at least at 25 microseconds. 45 nanometers is the size of the molecule, the typical size of the molecule anyway. So the median diameter. Now another final metric. We want to kind of quantify this aggregation. We're going to borrow an idea from computational geometry, Voronoi diagram. So you partition a plane here in such a way that each point inside this region is closer to this point than any other flat point. So if we try to, do the same thing in 3 dimensions, and we form these Voronoi cells. If we start from a uniform distribution, and over time we go to some aggregated system, right? Then we expect the distribution of these cell volumes, the volume of each region, to spread out. Like at first, they are centered around some value because it's a pretty uniform distribution. Remember how the simulation started. But over time, because some molecules come closer to each other while others are further away from each other, we expect this distribution to get higher and higher. So, or we're going to use either the JS divergence to measure this, quantify this aggregation, or the width. 90th minus 10 percentile. And in both cases, we see how it increases. So here you see 3 snapshots. I forgot to say this at 0, 15, and 30 microseconds. Okay, so this is what we have so far. And what we want to work on mostly is to study the effect of the alpha C domains. One thing I talked about earlier is about how the alpha C domains are swinging hands that capture other molecules. Well, we want to see what if our simulations can verify something that happens in experiments. What happens in experiments is that the lack of alpha C domains slows down this fiber assembly, which is not very unintuitive. And these results are more pronounced in more diluted solutions. Also makes sense. In more dense solutions with higher concentrations, the molecules will come closer to each other anyway. So their contribution is going to be less pronounced in more dilute solution. their role is going to be more pronounced in more dilute solution. So we want to quantify this effect and see if we can simulate it with coarse graining. So this is pretty much what we're working on in terms of this this project, this chapter. Second chapter is going to be shorter, don't be intimidated. So this is what we're doing in terms of fibrinogen and in terms of forward coarse graining. Now we're going to stop. We're going to put like a full stop, and I'm going to talk about inverse coarse graining and how we're trying to modify the recent methods in this field. So I guess we'll do questions at the end. So now we're going to talk about two-step backmapping of large protein. Again, I said it in the beginning. We have, the equivalent of image super resolution, taking a blurry image but making it clear. We do the same thing with molecules, starting from a CG resolution, going to a fine-grained resolution. Well, this is a challenging problem for multiple reasons. First of all, it's a stochastic inverse problem. It will be kind of like a bad thing if we had a model that only produces, a single output, because there could be multiple FG resolutions that map the same CG resolution. We just want it to be as realistic as possible. Yes.
Can I interrupt for a bit?
Yeah, of course.
So in terms of image super resolution, you would assume that you take information from the surrounding pixel and that adds information to the
Right.
pixel you're operating on
Yes.
to perform super resolution. In this case, I see it more like a micro and a macro scale.
Yes.
So what is the real parallel between the two words?
Well, I mean, it's again a matter of resolution and degrees of freedom. You go from few degrees of freedom to higher degrees of freedom, and you do want them to follow a distribution. You do want them to which brings me to the third point, I guess. You don't just want to learn how to, guess how a CG resolution maps to an FG resolution. You have to train a model that has a general idea about the topology of the molecules as a whole, so that you can make it as realistic as possible. Now, here, obviously, you don't have, pixels, so you don't have convolutions in the traditional sense, like in imaging. There's some graph-based algorithms that are used here. But that's a very good question.
So these smaller green dots, they become nodes in the bigger graph when you.
Yeah, so they will become something like this, or they should become something like this, hopefully, because that's what they represent.
Okay.
Okay? All right. So, and we also want a model that's very general, right? It maps different resolutions to the atomistic resolution. Now, some recent methods, some of them are energy-based. So, in one sentence, maybe we should have skipped it, random assignment of atoms, and then you run a simulation in the hopes that the actual, the all atom force field will restore the correct bonds, angles, dihedrals. So you put it randomly, and then you hope that a simulation is going to take care of it. This is very expensive though, because you have to run a simulation. If we want to do some data-driven methods, there's some deterministic ones with neural networks. Okay. Well, the problem with these is that they, as I said, they're deterministic. We want something stochastic, probabilistic. We want to learn a distribution and output a distribution. So that's why we have to use some sort of semi-supervised learning. We have CGANs, CVAEs, general adversarial networks, variational autoencoders, and diffusion models. And as I said, we're going to focus on large proteins for a reason, okay? But before we do so, very quickly, I'll talk about variational autoencoders. I'll skip a lot of it, I guess, because we don't have enough time to talk about everything. But the idea with an autoencoder, variational autoencoder, is that you have an input data. You try to map them to some sort of lower dimensional latent space, and then reconstruct them. The idea is that at the end of this forward passing and reconstruction and then backpropagation, you will learn how to form these neural networks and this latent space in a way that then you have a machine that creates these structures. I don't know how to put it in a better way. So by learning, it's as if I give you a Lego, I ask you to reconstruct it, break it down, and then put it back in. Well, at the end, you'll be good at putting back Legos, you know? You'll be good at creating Legos out of nothing, out of randomness, out of noise, okay? By disassembling it and assembling it again. So the loss, there is I'm not gonna go into the details of the derivation. It comes from variational inference. You want to maximize the evidence lower bound because maximizing this distribution is kind of intractable. So what you do is that you want to maximize the reconstruction and minimize the KL divergence between some distributions as a regularization loss. So like make the latent space a little bit better behaved. So this is what the loss in a variational autoencoder looks like. Very interesting, please. There's a lot of videos online, I guess, if you want to learn more. Conditional variational autoencoders, you add extra information to the network. You don't want to have unsupervised learning. You don't want someone to just create any sort of Legos out of noise. You want to give some extra information, make a farm out of Legos, make a spaceship out of Legos. This is the auxiliary variable, the conditional input. Over time, you know, learning from the data is gonna like take care of this labeling. So we're adding extra features to your generative network. Now, why am I saying all these? Because the reference model that we'll be using for our backmapping techniques do use conditional variational autoencoders. As a matter of fact, this model, CGVAE, is going to be our benchmark for multiple reasons. Number one, it's equivariant, probabilistic, not limited to condensed phase systems. It is more accurate than all of its predecessors, and unlike all of its successors, I would say, or I mean, at least all of them that I know of, it can have an arbitrary and customized CG resolution. So it doesn't start from a specific resolution. You can like say, oh, you know, I have a molecule, and we make a CG representation with 6 beads or 20 beads, or I don't know, another molecule, 5 beads, 3 beads, and learn how to backmap it. It will learn how to do it at various scales, various bead numbers. So it's very general. That's why we're using it as a benchmark model, because we do want something general. Yes.
What's the equivariance and symmetry group that you have?
Like translations and rotation. Yeah, I actually should have said this. Translations and rotations, because you want the operator, I guess, to like map the set, like translated structures into the similarly translated structures. So I'm not going to focus again too much on the derivation. It's very interesting. Again, you have to maximize an evidence lower bound. But now you know the log likelihood that you want to maximize is a conditional likelihood. Okay, and then you have multiple models, like an encoder, a decoder, an encoder, and a CG prior, all of which work together to map, you know, to take during inference a conditional input and give an estimated reconstruction. Okay, so CGVAE, the model that I showed you, and this model here, Gen Z Prot, they're both based on conditional variational autoencoders, and they're both implementing this strategy to map molecules of different resolutions back to their atomistic resolution. Now, we're going to be using this model, but it's not a benchmark model. It's very useful because it's more accurate than the previous one, but it's not general. The weakness here is that you can only start from residue-based coarse grain. If I take the protein, I can start from the alpha carbon traces. So it's very specific. It only starts with this. And it makes sense because, you know, residues, amino acids have similar structures. They're the building blocks of proteins. So if you train it on them, you know, it's as if you train it on all building blocks. So you can like generalize multiple proteins. And also, in order to implement this sort of like residue-based reconstruction, you have the physics-informed laws component to preserve bonds, angles, and dihedral. So we're going to be using this model somehow, but it's not going to be our benchmark. I guess, why am I so interested in this? This model has been tested on like small proteins, chignolin, alanine dipeptide, or large proteins with residue-based coarse graining. So what happens is that like the ratio between the number of atoms and the number of CG beads is kind of small. Most of them are in the orders of like up to 30. If we, the term I'm using here sometimes is the size of a CG bead. Size of a CG bead means the number of atoms corresponding to one bead. So as we can see, when we test this CGVAE model on proteins with a large CG bead size, so 60 up to 300, the reconstruction becomes a little bit worse, like from 2.7 all the way to 7.7, and now we're testing it with 5 beads, and I mean, I assume it's going to be a little bit worse, but we have to see. So the reconstruction RMSD, the deviation between the two structures, becomes larger and larger. So can we do better than this? Maybe we can. Well, first of all, here we show how like the model has been tested only on these. These are the CG bead sizes up to 30, and the values don't go beyond 2, pretty much. But then if we test it on larger CG bead sizes, the reconstruction RMSD becomes larger and larger. What we're going to do is going to do successive backmapping. So we're going to start with this heavy, very coarsened shape-based coarse graining. We're going to train a model that, like CGVAE, trained on the particular protein, not to reconstruct the whole thing in one step all the way to the all-atom resolution, but to restore the residue-based coarse graining, to restore the resolution of the alpha carbon traces. And then we're going to fine-tune a model similar to Gen-Z prop so that it can also reconstruct some these structures from their alpha carbon traces all the way to the all atom structure. So we introduce an intermediate step. Instead of like doing it in one step, we're going to do it in 2 steps. And this has multiple advantages. Well, the theoretical, I guess, reasons behind this is if you condition this reconstruction, then you take the log likelihood, then the evidence lower bound here is split into 2 steps. Remember earlier we wanted to maximize this by maximizing whatever was after the greater than or equal sign. Now we're going to do it in 2 steps. And notice, these 2 steps, they can be fully independent. You can have one resolution from here all the way from the fine grain all the way to shape-based coarse graining, and from fine grain all the way to residue-based coarse graining. So you can have 3 data sets instead of 2 with deterministic mapping. And what you're going to do is that you're going to split this optimization, this heavy optimization, into 2 steps. So you want to lighten the computational and memory demands. You might want to introduce variations to the 2 models. You want to explore the search space a little bit differently. They're not going to be exactly the same model. There's going to be variations. If there is an error in one of the 2 steps, you can identify where the error comes from. And because the search space is a bit reduced, you are less likely to be trapped into local minima. So that's another advantage of doing this stepwise. So we are testing this on a total of like 3,000 conformations of coming from an all-atom MD simulation of the protein EIF4E, the translation initiation factor. It's a protein with, the structure that we had has around 2,945, so 3,000, roughly 3,000 atoms. We're not talking about proteins with 20 atoms or 100 atoms. We're talking about 3,000 atoms. And we're using different CGB resolutions, like 10, 20, and 50. So these are the bead sizes. And with the one-step reconstruction, we get these values. With two-step reconstruction, we get much better values. So if we can see the third point, I guess, where the effect is more obvious, we see how, we start with some sort of hockey stick, and I expect this to become even worse, I guess, for lower resolutions because of these heavy computations. But we kind of flatten it. There is like a trend here, it gets higher, but at a much lower rate. So we take a tool that works very well, and by introducing this intermediate step, we lighten the optimization computationally, and we make it more practical for our purposes. So now we can use this tool for simulations with bigger proteins and few bead sizes, like fibrinogen that I showed you earlier. So now we try to turn this into a more practical tool. Obviously, we want to apply this to more and larger proteins. We want to introduce other intermediate steps. Maybe for larger proteins, another intermediate step is needed. Sometimes, there's a bit of a balance, in principle, you could introduce, 100 intermediate steps, but it might not be good. It's very computationally heavy. What's the point of doing it? So there is a balance to this. Then we also want to conduct an analysis on how this ratio, this bead size, CG bead size, affects the accuracy. And we also want to look at some other backmapping models, how we can modify them for to apply them to larger proteins with few CG beads. So there's some diffusion models that came after CGB and Genzik brought, but are mostly tested and used on residue level coarse graining or some Martini-like coarse graining. Very, very specific. So we want to generalize this as well. So this is pretty much it. So forward and inverse coarse graining. Thank you so much, and I'm happy to answer your questions. Before I guess I finish, I'd like to thank my advisor and also a bunch of people that helped me in this process, collaborators and the faculty from other departments like Professor Rafailovic from the Material Science Department. And yeah, thank you all.
Thank you, Yuger. It's a very nice presentation. Well, I'm shamelessly claiming that. Are there any questions or comments, or more questions or comments from the audience, online and offline?
Let's start with the question from Zoom, I guess.
Yes. Any Zoom questions? We have quite a few people on Zoom. Anyone?
Yes. Can I ask a question?
Yes, please.
Yes.
Okay, very, very interesting talk. Thank you. So I was just wondering if you were able to include any interaction between the solvent and your protein directly in your model, because that was not clear, because your prior includes interactions within the molecule you're trying to coarse grain.
Yes.
But
Yes.
what about
I
the
see
interaction?
what you mean. Yes. Yes. Obviously, it's a thought that has come to my mind, but I guess for what we did as a first step is to first train a model by itself. One thing I forgot to say is that the molecule was trained in vacuum. So thank you for pointing it out, I guess, that there is no protein solvent interactions. And the solvent was also trained by itself with DPD. And then we put these two together, matching the conditions that would be in a typical blood flow. but yes, it is a more obviously ambitious task to involve more information in the CG prior, but it's definitely something we are considering. Thank you.
Awesome. Thank you. Thank you. That's one clarification I had, I was seeking. Also, just a quick follow-up. What exactly is the multi-perceptron capturing on top of your pairwise interaction? Is that like the non-linearity? Is it like more of the many body effect, we would call it is that what you're trying to capture with that U-net term?
What part? Sorry, can you repeat that again?
So your U prior captures a lot of the pairwise interactions. I believe it's a Leonard-Jones potential. You have some,
Yeah.
angles. But what is U net exactly capturing? Is it like the many
Oh,
body
okay, yes,
effect?
yes. So because this prior is not like, it doesn't like capture 100% what happens like in the actual dynamics. We also have a scaled like black box implementation neural network matching this like these error terms. So we do still have a small component of the potential coming from like an uninterpretable source, but it's scaled accordingly so that it doesn't add too much. So matching the error terms, let's say.
Thank you. Yeah, any more questions from the from Zoom? No, you don't have to raise hands, just shout if you have any questions. Okay, if not, are there any in the audience?
Yes, in the room we have a question.
Yes, please. Oh, in the room, yes.
Yeah, thank you so much for this really nice talk. I actually have two questions. So one question, the first one, so you modeled the interaction between the different molecules with Coulomb forces, right?
Yes.
Do you know like what they are in reality? Like it's probably different ones, but is it like mostly van der Waals or is it cysteine?
So the literature says electrostatic, hydrophobic, but if you think about it, it's like it's a collection of interactions. It's not like between any two atoms. It's like it's a cumulative effect. Mostly hydrophobic or electrostatic in nature. Right?
Yeah, with hydrophobic you mean van der Waals, like London destruction?
Yes, yes, that too. Yeah, that too. I mean that would always be the case, right? London dispersion forces, like in any two.
The problems there are like aesthetic, which is very difficult to define them because like the other person had asked solvents and salts and modified.
Yeah.
We have to educate a guess.
Okay. So do you think you also have like a lot of like disulfur bonds?
A lot of what?
Like cysteine bonds there? Disulfur bonds?
I don't.
Okay.
Yeah.
Would you like to answer? Yeah.
Okay.
That's part of what we're trying to figure out, because he has to put the structure of the molecule online, and you found one structure, but apparently there's a lot of them, people are arguing.
Right, like if you go to like the protein data bank, there's different crystal structures for the same molecule that have been computed with different methodologies by different people. So yeah, but there's some like error that might be in including this.
I see, thank you. And the other question is like, how do you think your method compares to like Monte Carlo method? Because you also mentioned like you want something stochastic. So
For the backmapping, well, for backmapping, if you're saying Monte Carlo, I guess you're talking about this like random assignment and then trying to follow distribution. Actually, most of the literature I've seen has either these energy-based methods, so random assignment followed by a simulation, or data-driven methods using either simple neural networks that are very deterministic, or if you want to embed the stochasticity, SIGAN, CVAEs, and so on.
but you can also do that with Monte Carlo, right?
You can do that with Monte Carlo. Probably for small molecules, it could work. But if you have like a 3,000 atom protein, there's a lot of like degrees of freedom to consider with all the Monte Carlo simulations, right?
Yeah, but that's why I would think like Monte Carlo would be advantageous because it's like cheap if you increase the degrees of freedom and dimensionality.
Sure, yeah, maybe it's very heavy, I don't know, we'll see, yeah, it's, I didn't think about it, it's probably a very heavy computation, even with data driven, where you restrict your molecule to some sort of structures that are given in a data set, it still takes hours and hours, and sometimes, one thing I forgot to say is that the other methods, the previous methods, sometimes, when you have 3,000 atoms, the load is so heavy that even a 24 gigabyte GPU or 2 GPUs, I guess, with parallelization are not good enough to take care of the graph-based algorithms. Because graph algorithms, like they do store a lot of data in the meantime.
Yeah.
So we weren't even able to do that, like to run the molecule sometimes, to the model sometimes. Without some data shredding and model normalization.
Okay, thank you.
Yes.
Really awesome talk, thank you. I was wondering for the inverse coarse graining, when you were describing it in principle, you were mentioning how you want the model you're using to not just give you an individual answer, but to give you a distribution of answers.
Yes.
You could imagine going in your actual target system from 15 beads to 42,000 atoms. There's a lot of different possibilities for all
Yes.
these different groups that the beads are representing. But then when you are showing the validation of these sort of individual models, you're using individual like RMSD values. And
Yes.
so I was wondering if those are like averages for a distribution, or if there's some way for these models to weight the distributions that you get.
Yeah, so first of all, very good question. They are averages. And I wish we had shown you like error bars. But sometimes computations take a long time, training multiple models multiple times. So hopefully in like a few weeks or days, we can have the error bars as well. They're usually small, but at the same time, there's another metric that I didn't talk about because I guess I didn't have too much time that tries to quantify the sample diversity. So for a single molecule, you can get you backmap it multiple times. And you get a metric on how much of a variety of backmapped structures you get, in addition to how accurate they all are. So you want a very small RMSD for reconstruction accuracy, but you also want a relatively high sample diversity metric that actually takes this stochasticity into account. You don't want a single answer. And the semi-supervised methodology like CVAs does take this randomness into account from like in principle and in practice.
There's a hopefully quick follow-up then. If you have this ideal method that gives you very small average RMSD and very high structure diversity, would you take some, do you think you have any way to sort of weight that distribution to then understand like which of these structures is most representative of what you're actually would be seeing if you were able to do an all-atom simulation?
It would be nice, I guess, to see this, like sample it multiple times, backmap it multiple times, see the distribution of conformations. You know, you can reduce it, I guess, with some dimensionality reduction techniques to see which ones are like very similar to each other, how similar they are, which ones are like the energy minimum. Yes, that would be very nice to see, actually. Thank you for the suggestion, actually. And it would also be very nice to see, what was I gonna say? I forgot. Thank you for the suggestion. We will see this. One thing is that sometimes comparing it to fine-grained simulation data is very difficult because you have to run a lot of like MD simulations from
Yeah.
different initial conditions to like get a good representative sample of the conformational space that a protein can have. A limitation still of all atom simulations.
Of course,
Yeah,
thank you.
yeah, but thanks for the suggestion. Yeah,
Just a quick question. You've employed Voronoi tessellation to
Yeah.
track atomic volume. Out of curiosity, have you looked at specific indices as the system evolved in time?
no, I haven't.
I wouldn't expect a crazy amount of structure, but as it evolves, you see that kind of clustering. So I'm sort of curious about specific indices. As it
Yeah.
evolved.
Specific indices? No, not really, because one thing about the Voronoi tessellation is I do it at not at an atomic level, but at the molecule level. So the molecules, they all have pretty much the same like topology, even though they might have a different conformation. So if I would do it at an atomic resolution, then yeah, it would be very interesting to see actually which indices are aggregated together, which would be a good, like, I guess, indication of what atoms come closer to each other and they form bonds. But yeah, also very good suggestion. Thank you. Thank you. All right, anything else? Yes.
Okay,
Just one thing is like, so in your physics-based simulation, when you were, the physics-based model that you're using.
Mhm.
How do you decide how many variables you need to use? Were you just working off, okay, these are the forces I expect, or did you leave in space?
Yes, you do like, you have to make some assumptions, I guess, about the forces that you're gonna take. So we do have like some bonded and non-bonded interactions that are very like typically seen in proteins. But then that takes me back to the first question that I got. We had this black box implementation model as well that added some that added a little bit of that added like the error term, the noise term in the potential estimation. So here we have the U prior, but we also have this U net that's pretty much all the other interactions that we're like ignoring for now. So it tries to add up and balance it. But yeah. Yeah.
This is small follow-up, so it's like, so you made sure that your unit, whatever the neural net stuff was as small as possible. Right?
Sorry?
The error that whatever.
Oh, yeah.
You don't make-
This was scaled, I guess this is not a very good, I guess, representation. You wanted this to be scaled a little bit so that it only adds a small contribution to the whole potential estimation. Yeah, thanks for the question. There was a question online.
the question is actually thanking you.
Oh,
Yeah,
That's it. I just wanted to say
Yeah,
thank you
and
so
it's,
much for your presentation. Really
thank you.
appreciate your joining us today.
indeed. Thank you. And thank the whole audience for great questions and attention. Frankly, with the power of AI, we in applied math can tackle a problem as complicated as this. And on the other hand, your collaborations will be very much appreciated, including those good questions. Yeah, we'll hopefully, yes, indeed. Thank you, Yogurt, for a nice presentation, and good luck for the continued progress.
Thank you very much. Thank you.
Okay, bye-bye.
All right, if there are no other questions, I guess we can conclude for today.
Okay.
All right, okay, have a nice rest of the day, everyone. Thanks for coming. I really appreciate the questions and the participation. Thank you all.
Bye-bye.
Bye.